MCQs. Topic: Central Dogma of Molecular Biology, CDMB. 1. Who first proposed the central dogma of molecular biology? A. James Watson. B. Francis Crick. C. Rosalind Franklin. D. Frederick Sanger. Answer: B. Francis Crick. Explanation: Francis Crick proposed CDMB in 1958, later refined in 1970 to explain the unidirectional flow of genetic information. 2. The central dogma describes the flow of genetic information from A. Protein rightward arrow RNA rightward arrow DNA. B. DNA rightward arrow RNA rightward arrow protein. C. RNA rightward arrow DNA rightward arrow protein. D. DNA rightward arrow protein rightward arrow RNA. Answer: B. DNA rightward arrow RNA rightward arrow protein. Explanation: the classical dogma states that DNA is transcribed into RNA, which is translated into protein. 3. Which enzyme catalyzes DNA replication? A. RNA polymerase. B. DNA polymerase. C. Reverse transcriptase. D. Ligase. Answer: B. DNA polymerase. Explanation: DNA polymerase synthesizes a new DNA strand using a DNA template during replication. 4. Which process converts DNA information into RNA? A. Replication. B. Translation. C. Transcription. D. Reverse transcription. Answer: C. Transcription. Explanation: transcription is the synthesis of RNA from a DNA template using RNA polymerase. 5. The ribosome is primarily responsible for A. DNA replication. B. RNA splicing. C. Protein translation. D. Reverse transcription. Answer: C. Protein translation. Explanation: ribosomes decode mRNA into polypeptide chains using tRNA and rRNA. 6. Which enzyme is essential for transcription? A. RNA polymerase. B. DNA polymerase. C. Reverse transcriptase. D. Helicase. Answer: A. RNA polymerase. Explanation: RNA polymerase catalyzes the transcription of DNA into RNA. 7. Which of the following is an exception to the central dogma? A. DNA rightward arrow RNA rightward arrow protein. B. RNA rightward arrow DNA rightward arrow RNA rightward arrow protein. C. DNA rightward arrow DNA replication. D. DNA rightward arrow RNA transcription. Answer: B. RNA rightward arrow DNA rightward arrow RNA rightward arrow protein. Explanation: retroviruses use reverse transcriptase to convert RNA into DNA, an exception to classical CDMB. 8. Reverse transcription is catalyzed by A. RNA polymerase II. B. Reverse transcriptase. C. DNA ligase. D. Topoisomerase. Answer: B. Reverse transcriptase. Explanation: retroviruses like HIV use reverse transcriptase to synthesize DNA from RNA. 9. Which viruses replicate their RNA directly without DNA intermediates? A. Retroviruses. B. RNA viruses, example SARS-CoV-2. C. Bacteriophages. D. Adenoviruses. Answer: B. RNA viruses, example SARS-CoV-2. Explanation: RNA viruses use RNA-dependent RNA polymerase to replicate their RNA. 10. The expanded central dogma includes which unusual flow? A. DNA rightward arrow protein. B. Protein rightward arrow protein. C. DNA rightward arrow DNA. D. RNA rightward arrow RNA. Answer: B. Protein rightward arrow protein. Explanation: prions represent a protein-to-protein information transfer, misfolded proteins inducing misfolding. 11. Which of the following does not always lead to protein synthesis? A. mRNA. B. rRNA. C. tRNA. D. micrRNA. Answer: D. micrRNA. Explanation: non-coding RNAs, tRNA, rRNA, mRNA, siRNA, have regulatory/structural roles instead of coding for proteins. 12. Which step of the central dogma ensures genetic information inheritance? A. Replication. B. Transcription. C. Translation. D. Reverse transcription. Answer: A. Replication. Explanation: replication duplicates DNA to be passed on during cell division. 13. Mutations in DNA primarily affect which step? A. Replication. B. Transcription. C. Translation. D. Protein folding. Answer: B. Transcription. Explanation: mutations in DNA alter RNA transcripts, which may result in defective proteins. 14. In HIV replication, which sequence occurs? A. DNA rightward arrow RNA rightward arrow protein. B. RNA rightward arrow DNA rightward arrow RNA rightward arrow protein. C. DNA rightward arrow protein. D. RNA rightward arrow protein. Answer: B. RNA rightward arrow DNA rightward arrow RNA rightward arrow protein. Explanation: HIV uses reverse transcriptase to create DNA, which is integrated into host DNA, then transcribed back to RNA. 15. The enzyme that adds nucleotides during transcription is: A. DNA polymerase III. B. RNA polymerase. C. Primase. D. Ligase. Answer: B. RNA polymerase. Explanation: RNA polymerase reads the DNA template and synthesizes RNA. 16. Which discovery challenged the original central dogma? A. tRNA. B. Reverse transcriptase. C. DNA ligase. D. RNA splicing. Answer: B. Reverse transcriptase. Explanation: the discovery of reverse transcriptase in retroviruses showed RNA rightward arrow DNA flow. 17. Which type of RNA carries amino acids to the ribosome? A. mRNA. B. tRNA. C. rRNA. D. snRNA. Answer: B. tRNA. Explanation: transfer RNA, tRNA, carries amino acids to ribosomes for protein synthesis. 18. Which RNA acts as a template during translation? A. tRNA. B. rRNA. C. mRNA. D. mRNA. Answer: C. mRNA. Explanation: messenger RNA carries codons that ribosomes translate into protein. 19. The primary importance of CDMB in biotechnology is: A. Explains DNA structure. B. Predicts protein synthesis from genes. C. Describes cell division. D. Identifies lipid functions. Answer: B. Predicts protein synthesis from genes. Explanation: CDMB helps scientists predict how genetic information produces functional proteins. 20. Which of the following best explains prion diseases in CDMB context? A. Protein rightward arrow DNA. B. Protein rightward arrow protein misfolding. C. DNA rightward arrow RNA rightward arrow protein. D. RNA rightward arrow RNA replication. Answer: B. Protein rightward arrow protein misfolding. Explanation: prions replicate by inducing misfolding of normal proteins, bypassing nucleic acids. 21. Which scientists work with retroviruses demonstrated RNA rightward arrow DNA flow? A. Frederick Griffith. B. Howard Temin and David Baltimore. C. Oswald Avery. D. Erwin Chargaff. Answer: B. Howard Temin and David Baltimore. Explanation: they discovered reverse transcriptase in retroviruses, Nobel Prize 1975. 22. RNA viruses like influenza replicate by A. DNA polymerase. B. RNA-dependent RNA polymerase. C. RNA ligase. D. Helicase. Answer: B. RNA-dependent RNA polymerase. Explanation: RNA viruses replicate their genome using RNA-dependent RNA polymerase. 23. Which process is universal in both prokaryotes and eukaryotes? A. RNA editing. B. RNA splicing. C. Transcription and translation. D. Polyadenylation. Answer: C. Transcription and translation. Explanation: both prokaryotes and eukaryotes use transcription and translation, though regulation differs. 24. Which of the following is not part of the modern expanded central dogma? A. DNA rightward arrow DNA. B. RNA rightward arrow RNA. C. Protein rightward arrow DNA. D. Protein rightward arrow protein. Answer: C. Protein rightward arrow DNA. Explanation: no mechanism exists for proteins to directly encode DNA. 25. Why is central dogma considered the blueprint of life? A. Explains how carbohydrates form. B. Explains how genetic information flows to create function. C. Describes cell metabolism. D. Explains photosynthesis. Answer: B. Explains how genetic information flows to create function. Explanation: CDMB describes how DNA stores instructions that are expressed as proteins, enabling life processes.